Yes, madam. We are good to go.
Okay.
Good afternoon to one and all. I, Tanisha Sharma, student of BA Journalism and Mass Communication, am the host. We are delighted to welcome distinguished dignitaries, invited guests, faculty, staff, alumni, and students at the second Nanubhai Amin Memorial Lecture Series. This event will commence with the University Anthem. I request all to rise for the University Anthem.
विश्वामित्र के तट पे देखो शहर सयाजी नगरी है, माँ शारदा का मंदिर है, नवरचना यूनिवर्सिटी है। बनना है जो बन जाओ तुम, बात हमें बदलाती है, शिक्षण से ही स्वतंत्रता है, पाठ यही पढ़वाती है। नव नव रचना करते यहाँ पर छात्र और छात्राएं हैं, शिक्षण की खुशबू फैलाती नवनिर्मित शाखाएं हैं। प्रयोग से प्रेरित शिक्षा पर यहाँ जोर दिया जाता है, NUV का छात्र दशों दिशाओं में पर फैलाता है। निष्ठा तप और धैर्य यहाँ के शिक्षण की पहचाने हैं, शिक्षा और संशोधन से जो उन्नत राष्ट्र बनाते हैं। नवरचना नवरचना यूनिवर्सिटी, नवरचना नवरचना यूनिवर्सिटी, नवरचना नवरचना यूनिवर्सिटी, हमारी नवरचना नवरचना यूनिवर्सिटी।
Taking ahead this event, I invite Dr. Nilay Yagnik, Provost, Navrachana University, to throw light on Nanubhai Amin Lecture Series.
Thank you, Tanishka. On behalf of Navrachana University, I welcome all the distinguished guests, participants, faculty, students for attending the second Dr. Nanubhai Amin Memorial Lecture. I have my deep sense of gratitude to our president, Mr. Rahul Bhai Amin, and our chairperson, Mrs. Tejalben Amin, for joining us today and gracing the function with their presence. We started the Dr. Nanubhai Amin Memorial Lecture last year with a specific reason. We wanted to propagate the ideas that he had, the vision that he had, and the interest areas which he had. As you will see, many of the schools that we have in our university reflect his values and reflect his vision. He was a person with multiple interests, which included architecture, which included design, which included liberal studies, which included engineering, science, management, the legal profession, and of course, social work. So we have tried to see that in everything we do, we reflect his vision. And this particular lecture series is that way by which we connect to the outside world and do the things and spread awareness on the areas which he loved. So today we are indeed very honored to have with us Dr. Oliana Carnevali from the Polytechnic University of Marche in Italy. Dr. Oliana is joining us live from Italy. It's early in the morning for her. Thank you so much for joining us. Dr. Oliana is no stranger to Navrachana University. She has visited our university personally in February 2020, when we had organized the International Conference on Eco-Health and Environmental Sustainability. Dr. Oliana had delivered the valedictory address at that point in time. Dr. Oliana is a world-renowned researcher in the field of life sciences, environment, and human health. And that is the reason why we felt that it's extremely important for us to understand from her the kind of changes that are happening and what we should do to prevent such kind of contamination that is happening, which is affecting aquatic life, human life, and wildlife as a whole. Dr. Oliana has also been extremely helpful in enabling a collaboration with the Polytechnic University of Marke and Navrachna University. And I'm so happy to tell you that as a part of this collaboration, we are starting very soon, around December or so, 2 of our students will be going to Marke in Italy to do their internships. Initially, we are starting off in the field of life sciences, but later we plan to increase this across our university, the collaboration, which includes research and joint programs, conferences, seminars, joint curriculum development, joint sessions together. That's the basic idea. So thank you once again, Dr. Oliana Carnevali, and thank you all faculty, staff, students, and all the guests who are watching today. Thank you very much for joining us in the second Dr. Nanubhai Amin Memorial Lecture. Thank you.
Thank you, sir. The occasion today is marked by formalization of an important collaboration. I request Dr. Darshi Bakshi, Associate Professor and Program Chair, Division of Biomedical and Life Sciences, School of Science, to speak about the MOU.
Thank you, Tanisha. It gives us immense pride to announce to one and all present here the official declaration of Memorandum of Understanding between Polytechnic University of Marke, Italy, and Navrachna University, Vadodara. This MOU shall promote activities in the principal areas of student exchange, exchange of staff, collaborative research, instructional and cultural program, as well as exchange of research information. We are honored to have with us Professor Oliana Carnevali, who has been the key facilitator of this MOU. I would now like to formally request the permission from Professor Oliana Carnevali and our Provost, Dr. Nilay Yagnik, to announce and declare the MOU open.
Dr. Oliana, please do the honors.
Oh, yes, please.
Thank you, ma'am.
Thank you, Madam. Shri Rahul Amin, President, Navrachna University, is with us. May I invite you, sir, to offer your remarks at the event.
Good morning, Dr. Carnevali, and good afternoon to all the wonderful people who have joined this occasion, a significant occasion in the life of Navrachna University, where we have this MOU between our 2 organizations. We are indeed extremely grateful and honored, Dr. Carnevali, that you are here with us today, and that we will have many occasions to listen and hear your wisdom in the wonderful work you are doing in the field of marine environmental sciences, especially models of fish biology, because this is a very important field for us to understand. We normally associate environmental sciences with air pollution, etc. But you know that the marine environment and the marine pollution plays an equally important role, if not a better and more important role. And your work Dr. Carnevale is very significant and very amazing. I have read about it, and I would like to congratulate you for doing some most unique work, which I had never heard of. So welcome, thank you, and we are honored indeed. Today marks the 102nd birth anniversary of my father, Dr. Nanubhai Amin. He was an engineer, graduated from MIT, Boston, and Cornell University, Ithaca, New York, an electrical and mechanical engineer par excellence, an environmentalist, educationist, and a wonderful human being. He would have been so proud and so happy to listen to you today, Dr. Carnevale. And so I would once again welcome everyone, and we are all eagerly looking forward to hearing you and the fascinating work that you do, Dr. Carnevale. Thank you so much.
Thank you, sir. The second Nanubhai Amin lecture series has a distinguished speaker, Dr. Oliana Carnevale. I'm honored to introduce the speaker to the virtual audience. Dr. Oliana Carnevale is full professor of developmental biology at Polytechnic University of Marke. Her current research focuses on environmental risk monitoring with special focus on reproductive toxicology, as well as effect of contaminants on metabolic control and aging. She has more than 250 scientific papers to her credit. Her H-index is 50 and has almost 11,000 citations for her research articles. She is coordinating the doctoral courses as research deputy at Polytechnic University of Marke. She is the editor of 4 journals and president of master course in molecular and applied biology. Her scientific understanding on current status of environmental toxicants is of immense importance in current times when we are aware of the deteriorating environment around us that can be posing immediate health risks for the human population. Ma'am, welcome to Navrasha University. Dr. Oliana Carnevale will deliver the talk titled Contaminants in Water, Impact on Wild Organisms and Human Health. The talk will be followed by an interaction. I request the attendees to post the questions in chat. May I invite you, madam, to deliver the talk?
Thank you very much for this very nice introduction. And if I may, I want to share my screen with you. Can I?
Yes, please.
So dear colleagues and guests, I'm truly honored to deliver the second Nanubhai Amin Memorial Lecture organized by the Navrachan University. As was told previously, I was a guest of the Navrachan University during the second international conference in eco-health and environmental sustainability organized by the School of Sciences. I really appreciate the hospitality and professionalism of my colleagues, and I would like to thank the president of the university, Dr. Raul Amin, and his wife, Tajal Amin, that I had the pleasure to personally meet during the conference. That was my last conference in person before the pandemic, and I really have a very, very good memory of that conference. I also like to thank Professor Ramachandra and Professor Nalai Jajinik for this invite, very much appreciated. As was mentioned, so we just signed a memorandum of understanding between Navachamna University and Polytechnic University of Marke, and to establish a scientific collaboration of common interest. And we have several different areas that can collaborate together, and I am sure that we will have a mutual benefit from this agreement that allowed us to develop the research program, as well as the exchange of students and teachers. So I hope to see most of you in Ancona within this agreement that we just signed. And Ancona is, as you can see from, do you see my presentation?
No, not it.
No, ma'am. I think you may require to share it again.
Darshi, I think maybe you can share.
No, you are able to share. You will do.
You have to give Dr. Carnevali the screen sharing rights.
She's the co-host.
Yes, sir.
Okay.
We have tested it once and it was working.
Coming.
Okay, try again.
We are able to see it now, ma'am.
Yeah.
Okay, wonderful. So this is our campus, one of the 3 campuses we have at the Polytechnic University of Marche. Ancona is just in the middle of Italy, on the Adriatic Sea side. So the students, and we have already selected 2 of them, can join us in Ancona and enjoy the monument and the environment, especially as you can see here, the coast we have in our area. Regarding the memorial in honor of Mr. Nanubhai Amino, I have not had the pleasure to personally meet him, but I had the opportunity to learn how capable and visionary he was for what concerns renewable energy. I also know his sensitivity and commitment in the field of environmental protection. Somewhat very close to my heart, therefore totally shareable with him, as well as his passion for science. So my lecture, we will focus on contaminants in the water and their impact on wild and human health organisms. To start this lecture, I like to use something that was published a long time ago, but still very actual. So we can consider the current state of the universe as the effect of its past and the cause of its future. So the plastic contamination is really a big issue. So we can find plastic bottles from the bottom of the ocean to the top of the highest mountains, as you can see in these pictures. from Antarctic to Arctic. And in some cases, we can observe island made by plastics that we call ocean gyre. But we know that whatever goes in the ocean come back to us. So we are really concerning about this, because it is clear that the plastic additives used to bind the polymers are leached and can interfere with the endocrine system, acting as endocrine disruptors. So here is the classic definition of what it is an endocrine disruptor, which is a crucial point of my lecture. So it is something that is produced many times by the men, is an exogenous substances or a mixture, which is even worse, that can alter functions of the endocrine system. So the endocrine system is one of the most important systems the organism have, controlling all the physiological activities. And the alteration of the endocrine system might bring to adverse health effects in intact organism, or sometimes we have proof that can alter even its progeny or the entire population. So starting from this, we soon understand that such contaminants can be really dangerous for the organism in general, including human. So study performed on contaminants have allowed us to classify the contaminants as endocrine disruptors. And these substances can be divided in different classes, like estrogenic or anti-estrogenic substances that can work as estrogen-like substances. Estrogen are the signals mainly responsible for the female reproductive activity. And there are other substances that are defined anti-estrogenic, so can contrast the effect of endogenous estrogen and interact with a number of physiological processes. Or can be anti-androgenic. Androgen are the main hormone involved in male reproduction. So if we have substances that can interfere with the endogenous androgen, it can interfere with reproduction of men. And then we have progestogens. Usually they are used as contraceptives, but we can find these substances in the environment. How do work the endocrine disruptors like plasticizer? They are the similar function of hormone, and so they can bind with the receptor. Here you can see a number of receptors that have been proven to bind plasticizers, and these receptors are linked with reproductive process, for instance, the one I'm indicating, or with the metabolism, the rest. And what happens when this contaminant bind the receptor? So the response of a cell is altered. All the cells that contains these different receptors, their function is altered. And this can bring to some physiological problem. Many of these contaminants have been tested in organism models. Here you can see zebrafish, which is considered one of the most important model for toxicological studies. Here we can see some results. I'm not going too deeply the results because I just want to give a general idea how we can say if some product, some chemical products can be dangerous or not. So we use zebrafish, male and females, see the effect of new phthalates, the isononyl phthalates. These phthalates have been used recently to substitute another phthalates, the diethylhexyl phthalates, that was proved to be very toxic for organisms, included human. So our aim was to see whether DNP was safe or not. And we analyzed the effect of these environmental contaminants in male and female to see whether the reproductive physiology was affected or not, but as well as the metabolism. Generally speaking, we can see that the dimension of the gonads was affected. So both testes and ovary were significantly smaller in animals exposed in a chronic way. So in this case, the contaminants was added in the water. Different doses have been tested, and now I will explain why. And we could see the reduction of the gonads in male and female in a significant way. When we move to the distribution of the different class of oocytes, since the maturation of the ovary takes some time and the dimension of oocyte is changing during the gametogenesis, and we found that the distribution of these different grade of maturity of oocyte were affected. But please look at this graph where the number of eggs, it means fecundity, was significantly lowered. And what we can see, the decrease of number of eggs is not dose related. So we don't have the effect we can see is dose specific. And this is very important because we cannot test just one dose, but we should test at least 5 different doses because, as you can see, the lowest and the highest dose in this case give exactly the same results. Why other doses have less detrimental effect? So it is very important to test different doses because sometimes lower doses are the most detrimental. Similar effects were observed with different contaminants that is very much spread around the world, the bisphenol A, and we can see that also this contaminant can be considered very dangerous for, in this case, we can see testes, and the dimension of testes and the spermatogonia number was significantly down-regulated. Similar study have been performed in my laboratory in marine species to test the effect of these contaminants in the ocean, in organisms living in seawater, because sometimes the behavior of contaminants in seawater is different respect to the behavior in fresh water. The results we obtained in terms of effect on reproduction was really impressive. Sea bream is an hermaphrodite species that passed the first 2 years of his life as male and then naturally become female. So we use sea bream during the male phase, and we test the effect of these contaminants that was used to substitute different compounds like the diethylhexyl phthalate, as I mentioned before. So this phthalate has a very detrimental effect, probably similar to the compounds that have been substituted. In fact, we could see testis regression. The quality of sperm was significantly affected, and the sperm velocity was reduced. But also the steroidogenesis was altered when these organisms were, in this case, fed with diet contaminated with this phthalate. Here you can see the reduction of one of the most important hormone for fish reproduction in male, the 11-ketotestosterone. And similar effects have been found also with BPA in the same model. And as you can see, the BPA was able to reduce the higher level of this contaminant, the level of 11-ketotestosterone, and concomitantly, we could find an increase of estradiol. Estradiol is a typical hormone in female, so it seems to have a feminizing effect in this species. Also, the quality of sperm was affected, and the motility duration, the time in which the sperm is moving, was significantly reduced by the higher dose of BPA. So these results were associated with a number of changes in gene expression as reported in the heat map. And we can see here a long list of genes that were down or up regulated by the exposure to this contaminant. And you can see here that the genes are involved in both metabolism process or reproduction. Another clear effect of these two plasticizers is about their ability to induce the death of cells at ovary level. So we call this kind of death atresia or apoptosis. And when the animals, the female, were exposed to BPA, you can see here the increase of atretic follicles in the gonads, in the ovary. So this was a proof that BPA could affect the fertility, since could induce atresia at ovary level. Similar results were recently observed with the DNP in the same animal model, the zebrafish. And in this case, we use different methods. Before we just focused on histology. Here we search for a specific sign. Here you can see tunnel analysis that evident the dead cells in the follicles. And again, here the immunolocalization of a sign like caspase-3 in the follicles. This is a typical sign of apoptosis, a programmed cell death that occur in the ovary of zebrafish female exposed to DNP via water. We are even more worrying about the effect of this plasticizer because we demonstrated the transgenerational effects of the chronic exposure to BPA. In our study, we studied the transgenerational effect via mother. It means that a female that we call F0, first generation 0, we expose this female to BPA, to an environmental dose that did not affect really the fecundity. Okay, because when we expose a female to 5 microliter of BPA, reproduction was blocked, so we could not make any additional studies. Well, we decided to use this environmental dose to see what could be the transgenerational effect via mother. So these females were crossed with control male, not exposed to any contaminants. And then we got the first generation of embryo. Part of them were analyzed, part were grown, and we got the female F2. So the children of the mother exposed to this contaminant. And these females were used to make the second generation of embryo, crossing them with the control male. And we analyzed the F2 embryos as well. And we checked the fertility of F2 generation females, as well as embryos quality in the 3rd generation. So what we found was that the gonads was significantly reduced in F2 generation, as well as fertility. And when we search the quality of embryos F1, F2, and F3, we could find exactly the same alteration in all the generation we analyzed. So this was a clear proof that BPA could transgenerationally affect these organisms, at least zebrafish, and give some alteration by epigenetic changes in the embryos. So this is something that let us to think about, since zebrafish is a wonderful model also to predict what can happen in a human, since the genome of this model has a very, very high synteny with human. So we can translate the results we obtain in zebrafish in human, and maybe use the same bioindicators in human to see if something is changing, or in some mammalian, other mammalian model like rat or mouse. And this brings us to have an overview what is happening in human. So human fertility rate is decreasing in many parts of the world. So the total fertility rate tell us how many children for couple is typical in the different countries. So the very low TFR is typical of Western Europe and China, while very high TFR can be observed in part of Africa, as you can see here. So Indian has a good average of TFR, but is significantly decreased respect to previous data regarding, for instance, 20 years ago. So Italy has a very, very low TFR. So this is the numbers related to Indian. So in 1919, the TFR was 4, and actually is 2.28. And in your region is in Gujarat, the ratio is very similar to the national one, is 2.2. Look, in Italy, we have 1.27, which is really very, very low rate that indicate that the population is becoming older and older. And this is, of course, not very good. So what can be the possible cause for female infertility? There are many reasons, like hormonal problems, some syndrome like polycystic ovarian syndrome, endometriosis, which is a kind of inflammation of the uterus in human, in female human, obesity, but also some genetic problem, and the presence of endocrine disruptor chemicals. And I want to add that many times this kind of problem can be caused by endocrine disruptors like obesity and endometriosis, or this PCOS syndrome. And even as we have seen in our models, the alteration of hormone secretion can be caused by endocrine disruptor. And but actually we can see that people is more careful in the selection of products. And if you go to the supermarket, more and more often we can see products reporting BPA free or phthalate free, that are exactly the contaminants that we are studying in our lab and many labs in the world. So people is concerning about the presence of these substances because they know that can be dangerous. However, the substitute sometimes used when they put this label, I'm not sure at all. So this something that we have to consider, and the researcher should make research at this level to see whether the new products make people safe or not. But where we found these contaminants, so we can be exposed every day, just drinking water or eating canned food or food in plastic containers, or with product for our personal care or cosmetics, or for cleaning our house or cooking in non-sticking cookware. So these are the most typical sources we can find. And how is the mode of exposure? The mode of exposure can be very different. Dermal exposure for cosmetic or personal care substances, or might be with wearing specific clothes that contains retardant of flames or other products that are defined endocrine disruptor already. And then inhalation, or by food with the diet, or during the embryonic and fetal life, we can be exposed to these contaminants. And, we can see the effects even in the future generations. This is an example of contamination during fetal life. So in the amniotic fluid and in the blood of umbilical cord, and we can find a number of contaminants. Here are listed some of them. And in the pregnant woman, have been analyzed the presence of phthalates or flame retardants, and as you can see here, 99% of women present these compounds in their urine or blood. So, and again, in the umbilical cord and blood, over 200 toxic chemicals have been identified. So, and to say how is diffused the presence of these contaminants, the analysis of BPA in the American population find the presence in the organic fluid of these contaminants in 9 out of 10 people. So this is very, very general problem. what can be the effects of the presence of these contaminants in our children? So there is a relation between the presence of these contaminants and alteration of behavior and memory. Like BPA and phthalates have been associated with the attention deficit and hyperactivity disorder and emotional problems. In my experience along the year, I can see that the number of people affected by ADHD certified by the physician is increasing in a really impressive way. So many students are asking for considered ADHD and have a different exams because they are having problem in the attention deficit. Okay. As I mentioned before, these substances have been clearly associated with metabolic alterations. So this was clearly evidenced in models, and from the study in my laboratory, we found that both BPA and DNP in different models, sea bream and zebrafish, had an effect on metabolic alteration. And in addition, we found for the first time that these contaminants were able to alter the endocannabinoid system. So I don't know how many of you know this system, but I can tell you that is a very important system working on the control of metabolism, reproduction, immune system, and many other activities. So the effect of these contaminants on this system is increasing the concern of the effect. Probably we know some effects, but not all the alteration that can be caused by these contaminants. And at the end of our study, we found that both BPA and these new phthalates was able to induce steatosis in the liver, and this was associated with a number of genes involved in metabolism, as you can see here, that as shown in this heat map, were up or down regulated by the exposure to these contaminants. Similar effects have been observed also in human, and exposure to childhood to endocrine disruptor can induce obesity. In fact, the prenatal exposure to these contaminants are able to reprogram the adipogenesis. So the number of adipose cells can be higher when the children are being exposed during prenatal life. So this is associated with a number of contaminants. Here you can see the list of contaminants that have been observed in the urine or blood, and these are the contaminants, the source of contaminants in the everyday life. So most of the time can be due to food packaging. The prenatal exposure can also alter the birth weight.
And somebody, can you please? I'm so sorry, Dr. Xavier. I think you're unmuted. Please mute yourself. Everybody except for Dr. Oliana. Otherwise, it's very disturbing for her. Thank you. I'm so sorry, ma'am. Please continue.
Okay, thank you. No problem. So it can alter the weight at birth. So in some cases, we can see an increase of birth weight, like with some surfactant and phthalates. We can see the weight of the bird is significantly higher. In some other case, we can have an alteration of intrauterine growth that is consists in a restriction of growth. So the presence of these contaminants have been associated with a number of pathologies that are listed here in this slide, and in particular in men, cryptorchidism of hypospadias have been associated with the presence of endocrine disruptors, but also the increase of abortion or ectopic pregnancy. So this is something that affect the mother, have been linked with the high presence of these components, including the neurobehavioral disorder that I mentioned before. Another very hot topic is related with the presence of microplastic. So these contaminants have been found in many marine organisms and also in many human tissue. So our question was, can microplastic cross the placenta? And the replies, unfortunately, is yes, they can. So we sampled two different parts of the placenta, the maternal side and the fetal side. And we found microplastic in both of them, as well as in the chorionic membranes. So here, the method used to identify microplastic, and we use the Raman spectra, and then this analysis to identify exactly the type of plastic was done using the spectral library. And for some of them, it was possible to identify the polymer, the original polymer that is the polypropylene. For others, we were unable to say what was the original polymer. So the last part of my talk is about how to mitigate the toxic effect of endocrine disruptors. So one of the first evidence were published in the 2020 from colleagues from China, and they tested another perfluorobutansulfonate that were used to replace a very toxic PFOS, and they make a shorter chain, and they substitute the carbonate with these new compounds. But again, the substitute was toxic as well. And these colleagues found that the PFBS was able to alter the microbiome composition in the gut of fish exposed to these contaminants. So these contaminants can alter the composition of bacteria at gut level. And this is very, important to know, because the microbiome is very important for regulation of metabolism pathway. So this Chinese colleagues, the group of Chen, found that the introduction of probiotic, so a benefic bacteria, could modulate the lipid disorder induced by these PFBS contaminant. So this was the first evidence. And the results are not totally satisfying, because the Lactobacillus rhamnosus, the probiotic, was used to mitigate the effect of these contaminants. The cholesterol level in the blood was reduced, and the PFBS alteration of bile acidic metabolism was reduced, but at the same time there was an increase of triglyceride. It means that there was higher steatosis. So maybe Lactobacillus rhamnosus is good for cholesterol and for bile acidic metabolism, but not for triglyceride. So additional studies are needed to really clarify the role of a probiotic in the mitigation of these contaminants. Similar results have been obtained with another kind of probiotic in our laboratory. We used a mix of probiotics, 7 different strains, to mitigate the effect of BPA and reduce the toxicity in zebrafish. And we found a very nice sex difference in reply to this administration, but in male, we found a mitigation of the negative effect of BPA. So, of course, additional studies are running actually, and we are testing the effect on metabolism as well, as was done by our colleagues in China. So this is my last slide, and I really like the sentence used in the brochure of this event. And I think every researcher should continue to be inspired by person by like Nanubhai Amin, and our role is to spread the light of knowledge in the world. So, and the message I want to you take home is, so we have clear results for suggest that the production of new chemical and the use of substitute should include endocrine-based tests. This to guarantee their safety, as well as facilitate a progressive transition toward green chemistry. This is something that we really want if we want to protect the environment. So, and this will allow us to provide info for environmental friendly products and to improve the sustainability. So this is my last message. And of course, less plastic, more future. And I like to thank people that collaborate with my laboratory. Here we can see the PhD and postdoc that used to work in this field, especially Isabel Forner, Francesca Madonna, and Danilo Basili, and all the colleagues around the world that collaborate with us. But I also want to share, to thank Dr. Winkins for sharing some picture related with Indian condition and others, for supporting me to prepare this PowerPoint presentation. So, and thank you all for your attention.
Thank you, ma'am. The session is now open for an interaction. I request Mr. Rushid Dubal, Assistant Professor, School of Business and Law, to coordinate the interaction.
Audible? Hello? Yeah, I hope I'm audible.
Yes.
Yeah, thank you, ma'am. Thank you for such an enlightening session. I will continue with the question and answer session. The first question from Icha Sharma, SYBSc student. What if the water gets contaminated by pesticides? What harm can this type of contamination do to women's fertility?
Dr. Oliana, SY means second year, Bachelor of Science student.
Pesticides or other contaminants that we are worrying a lot, because it is clear their ability to bind with thyroid hormones, for instance. And thyroid is connected with reproductive physiology as well. So yes, also pesticides can alter reproduction. I'm not involved in studies this kind of contaminants, but I have read around that, yes, it can be dangerous for gametogenesis and general fitness, reproductive fitness.
Okay. There is one more question from a student, Mihina Pradhan, BSc Micro student. How were the contaminants able to alter the metabolic system?
How they can you repeat how the contaminants?
Yeah. The question is, how were the contaminants able to alter the metabolic system?
Okay, the metabolic system is very complex, so we didn't have time to go in deep in this specific task, but there are a number of enzyme and system, like I just talked about, endocannabinoid system that can control food intake and metabolism. So many contaminants can alter the transcription of genes are involved in the production of a specific enzyme involved in the lipid accumulation or lipid metabolism or triglyceride, as we have seen. So there are really a quite large number of markers indicating that the pathway, metabolic pathway, can be altered at cellular and physiological level.
Okay. There is one.
Okay, I can see an answer raised.
Rushit sir, can I ask a question?
Oh yes, please.
Yeah, buongiorno, Professor Carnevale.
More
Buongiorno.
question from Dr. Parth Pandya. Shall I repeat the question, ma'am?
Yes,
Yeah.
please do.
Yes, one more question from Dr. Parth Pandya. The question is, what should be the precautions to take if these chemicals are getting used day by day?
Oh yes. We have to select the product we are using every day. So we know that some food can be contaminated because they are in a plastic box or in can, and there are some chemicals like parabens that are inside the personal care products. And actually, there are, some products labeled with paraben-free because paraben have been identified as endocrine disruptors. So, and actually, they've been excluded from these products. But I want to move the attention not just as consumers, but I think we need collaboration with the chemical industry. And your district is very important, if I'm correct, because the chemical industry is very strong. So what we need is to collaborate with the chemical industry and to test the compounds they are releasing before to find them on the market. So this is the real message. I mean, we have to bring to the consumer, to the society, and sensibilize the producer to care about the health problem for wild organisms, but also for human. So this is very important because we know that it is very, very difficult to, eliminate from the water these contaminants. So any water treatment plant is able to really eliminate these endocrine compounds from the water. So we are recycling water, and so the concentration of these substances in many cases is increasing.
Okay. There is one more question from Dr. Sandeep Patil.
Please.
Even if we use BPA-free or DINP-free plastics, we shall place them in refrigerator or regular basis. Will the refrigerant can also be a cause of endocrine disruptor?
I'm sorry, I don't know exactly the way of conservation, if the way of conservation provide more or less release of contaminants. For sure, both bags used for cooking, which is become popular as well, at least in Italy, with some food that need just to be warmed up before consuming it, or to refrigerate the food. In all cases, we can find contaminants in the food, especially with the kind of food rich in fat. So this contaminant can easily pass from the package to the food. I'm sorry not being able to be more precise, but in general, yes, even if we fridge the food in plastic box, part of the plasticizer can be released.
Okay. There is a question from Navya Dubey, who is a BSc microbiology student. The question is, how can contaminants be reduced on a larger scale, like in oceans, because pollution in them directly and indirectly affects the humans through food chains?
This is the big problem. So we actually do not have technologies to solve this problem. So that's why we, as a consumer, we should select objects and food possibly free from these contaminants. But there are some new technology actually that are still at the experimental phase, so they are not commercially viable, that try to decontaminate the ocean from the plastic. But in this case, the plastic are, treated at very high temperature, and they do not release any toxic and like dioxins and so on. But the problem is the plastic in the ocean is releasing the plasticizer. So, and we have in the water not just plastic bottles or whatever, but we have these chemicals free, and this can be part of the diet of our wild organism, and then it's enter in the food chain reaching human as well.
Okay. There is a question from a faculty, Tejal Gajaria. Possible mechanisms by which probiotics playing role in amelioration.
Okay. We don't know exactly which pathway. We are just at the beginning, but we know that many of these contaminants can cause the dysbiosis in the organism. It means that the content of the type of a microbiota is altered by the presence of these contaminants. And one of the first approach is to include in the diet some species to solve the dysbiosis problem. So the idea is to have a very nice sorry, to have a very nice microbiome that can work well with the metabolism. But we know that some of these bacteria play a very crucial role in metabolism. So we did a lot of study on this, and we could see that the lipid metabolism can be positively affected by the presence of specific bacteria. So the idea is to have a better microbiome and guarantee its functionality, and so contrast in this way the dysbiosis that was caused by the contaminants.
Okay. There is one more question from Associate Professor Neha Sarwate. How can the treatment plants in cities be designed and modified to address the contaminants of urban lifestyle?
Can you repeat the question?
Yeah,
please?
sure. The question is, how can the treatment plants in cities be designed and modified to address the contaminants of urban lifestyle?
Okay. I'm not an engineering, I'm not an expert of it, but there are several different approaches. Maybe none of them totally satisfy the request to clean the water, but as far I know, there are different possibilities to act on the treatment of water, but still I don't think we have the technologies to have clean water at the end. But they are improving a lot. So I'm sorry, I'm not, as I told you, involved in this kind of approach, but as far I know, more and more people are working on this.
Okay.
You've gone mute. Rushit, you are muted.
Okay, disconnected for a minute. I hope I'm audible, sir.
Yes.
Yeah. The question from Hitar sir is, media play an important role in taking such detailed research work to masses. How the media is highlighting scientific research in Italy? Does it reach to masses in a right manner? And does it result in pushing the government to take action in reducing the usage of plastic?
This is the crucial point. So I totally agree that media are very important to tell the consumers, but also the people taking decision that there is a lot of concern from the scientific community regarding the use of specific chemicals. But this is not an easy task, even if for several contaminants, there are clear evidence about the negative effects. I'm speaking about the European community, for instance. Still, after many years, these chemicals are not banned yet, okay? So, but if the consumer is concerning and select product with without these compounds. So that's the best answer to the problem, because in this case, the chemical industry, is forced to select better compounds. Of course, the compounds need to give the same performance, but if we can support them, we are available for that. We can select the less dangerous compounds. This is something that is actually not well done or not enough done, but together with the capacity to divulgate what are the experimental data available so far. I think the consumers become more and more responsible, and they will really contribute to the reduction of the impact of many substances we are using actually, without know that may be dangerous.
Oh, okay. So here is the last question for this forum by Associate Professor Neha Sarvate. The question is, are there any specific microbes, floral and faunal species that help mitigate the key contaminants?
Oh, okay. The study at this regard are very limited, but I can see that Lactobacillus rhamnosus is the strain, but is not the only one, was able to reduce the cholesterol in zebrafish exposed to contaminants, and we use a mix of different probiotic like Lactobacillus acidophilus or Paracasei or others. So in this case, we also see a clear effect on the reproductive activity, but we are actually analyzing the results on metabolism in terms of metabolome and metagenome. So how these contaminants, in our case BPA, can affect. We repair this damage giving a mix of probiotics and see what happens. So, and these preliminary results tell us that really we can mitigate the effect of BPA in terms of metabolism and reproduction. So it is just the beginning, and it would be very nice to have more and more research group working on this aspect, because this is what really important. So if we cannot avoid these substances from our day life, so but we can mitigate it, just assuming some probiotics, that would be wonderful. So but we need deeper studies to really see if it is good or not, because as I mentioned, we can have a lower level of cholesterol, but then we have a higher level of triglyceride. So what is better? I don't know. So but we should look for something that really can mitigate most of the most important pathway and solve the problem. But this is just the beginning of the story, and probably in the next year we will have more knowledge at this regard.
Yeah. Thank you for sharing your thoughts, ma'am, and specifically answering all the questions, which is very interdisciplinary in nature. The questions are coming from architecture to mass media to other aspect, and we're really glad to hear your views about many multiple aspects of the same topic. I would like to hand over to Tanisha. Tanisha?
Thank you, sir.
Yeah, please take it forward.
To mark a formal closure of the program, it is appropriate to propose a formal vote of thanks. I invite Dr. Sandeep Patil, Associate Professor and Program Chair, Chemistry, and Head, University Research Cell, to deliver vote of thanks.
Thank you, Tanisha. I am privileged to offer a vote of thanks at this prestigious event, Sri Nanubhai Amin Memorial Lecture Series. On behalf of Navrachna University, Vadodara, I sincerely thank Dr. Uliana Carnevale for kindly accepting our invitation to deliver the second Nanubhai Amin Memorial Lecture. Madam, your rich insights as shared today with a wide audience from different sectors will help to continue the legacy of Sri Nanubhai Amin as he continuously strived for dissemination of knowledge and support for science, technology, and environment. We express our gratitude to Sri Rahul Amin, President, Navrachna University, for the remarks shared. We are thankful to Srimati Tejal Amin, Chairperson, Navrachna Education Society, for her gracious presence at the event. Dr. Nilay Yagnik, Provost, Navrachna University, has conceptualized this lecture series. Thank you, sir, for offering this platform and for being with us today. The event of such an immense stature would not have been possible without the strong support and guidance of Dr. Sandeep Vasant, Registrar, Navrachna University. Thank you, sir. We thank Dr. A.V. Ramchandran, Mentor and Professor Emeritus, School of Science. Professor Ramchandran has facilitated this collaboration. Sir, we acknowledge your efforts for the help in having Professor Uliana Carnevale as the distinguished speaker for this prestigious event. I also appreciate on behalf of Navrachna University the presence of all the dignitaries, invited guests, faculty, students, and all attendees. Thank you for being with us today. I also take the opportunity to thank the administrative staff, the technical staff of Navrachna University, especially Mr. Shahid and Mr. Sagar for helping us in hosting the event. I appreciate and acknowledge all the participants for the presence and precious time that you have spent with us. Thank you once again. Tanisha, please take forward.
This brings us to the end of the program. I request all to rise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
I once again thank all for joining us at the event. Wishing all a pleasant day ahead.
Thank
Thank
you,
you,
Professor
Dr.
Oliana, very much.
you. Thank you.
Thank you, Dr. Oliana. Thank you.
Thank you to you. It was really an honor for me.
Pleasure is
Thanks
all
a
ours.
lot. And we stay in touch with at least some of you
Yes.
Yes, ma'am.
to have the possibility to collaborate with me or with my colleagues at the University of Polytechnic of Marques. So please contact me, and even for, very formal question, and then I will be able to support you or ask the person in charge to do that.
Dr. Oliana, we also have a strong team of faculty in computer science and technology. We are also looking forward to have some sort of collaboration with our technology people as well.
Yes, that is very important point also for me,
Shahid.
because